Hello everyone, welcome back to my channel. In this video, we're going to talk about replication. Now before we start, if you are new here, please make sure that you hit the subscribe button so that you get notified whenever I post a new video. And if you find our videos helpful, please do not forget to hit the like button. Both these things help us a lot. So in the last video, we talked about DNA replication in bacteria. In this video, we're going to talk about DNA replication in eukaryotes. As mentioned in the last video, DNA replication is semi-conservative. It was proven by the Michelson and Stahl experiment. You can watch the last video for more details. So, there are three main steps of DNA replication. The first step is unwinding or separation of the two strands. The second step is the formation of the new strand by adding nucleotides complementary to the template strand. And the third step is the joining of the Okazaki fragments. DNA replication occurs in two strands, synthesized differently due to the anti-parallel nature of DNA and the 5e3 synthesis limitation of DNA polymerase. Leading strand, DNA polymerase synthesizes DNA continuously in the 5'3" direction. Lagging strand. DNA is synthesized discontinuously as Okazaki fragments opposite to fork, synthesized in small 5 and 3 pieces, each starting with an RNA primer. In the leading strand, primers are still required, but only once at the beginning, near the origin of replication. Now, unwinding of the double helix involves several proteins. The first protein is helicase. Helicase uses ATP to unwind the double helix and separate the two strands. Now, once the double helix is opened up, there will be a Y-shaped structure formed which is known as the replication fork. At the replication fork, the two strands are held apart by single-strand binding proteins. There is also another enzyme present at the replication fork called topoisomerase. Now, topoisomerase is an enzyme that relieves the strain of supercoiling ahead of the replication fork. This is because when helicase unwinds the DNA gets supercoiled ahead of the replication fork, and it becomes difficult for helicase to continue unwinding the DNA. So, to relieve this strain, topoisomerase makes a cut in one of the strands, allows the strands to swivel, and then rejoins the cut strands. All right, now the next step is the formation of the new strand by adding nucleotides complementary to the template strand. For this, we need an enzyme called DNA polymerase. DNA polymerase synthesizes a new strand by adding nucleotides to the 3' end of the growing strand. Remember, DNA polymerases can only add nucleotides to an existing strand. They cannot initiate the formation of a new strand. So, for the initiation of synthesis, we need a primer. A primer is a short RNA sequence that is complementary to the template strand, and it provides a starting point for DNA polymerase. The enzyme responsible for making the primer is primase. Primase is an RNA polymerase. So, what happens is, first DNA is unwound, and a primer is made by primase, and then DNA polymerase starts adding nucleotides to the 3' end of the primer. DNA polymerase reads the template strand in the 3' to 5' direction and synthesizes the new strand in the 5' to 3' direction. Remember the central dogma, DNA to RNA to protein. So if the template strand is in the 3' to 5' direction, the new strand will be synthesized in the 5' to 3' direction. This is because new nucleotides are always added to the 3' end of the growing strand. Now, since DNA polymerase can only synthesize the new strand in the 5' to 3' direction, but the two strands of DNA are anti-parallel, this means that DNA polymerase can only synthesize the new strand continuously on one template strand. On the other template strand, DNA polymerase has to synthesize the new strand in short fragments. These fragments are called Okazaki fragments. Each Okazaki fragment needs its own primer. So the process is as follows. First, primase synthesizes a short RNA primer on the lagging strand. Then DNA polymerase extends the primer by adding nucleotides to the 3' end of the growing strand. When the RNA primer is almost at the end, another RNA primer is made by primase, and DNA polymerase continues extending the strand. This process continues until the entire strand is synthesized. After the RNA primers are made, they are removed by RNase H enzyme and replaced by DNA polymerase. Finally, the gaps between the adjacent Okazaki fragments are sealed by the enzyme DNA ligase. So, to summarize, in the first step, DNA is unwound by helicase. Single-strand binding proteins hold the two strands apart. Topoisomerase relieves the supercoiling ahead of the replication fork. Primase synthesizes a short RNA primer. DNA polymerase synthesizes the new strand in the 5' to 3' direction, using the template strand as a template. In the second step, on the lagging strand, DNA polymerase synthesizes the new strand in short fragments called Okazaki fragments. Each Okazaki fragment needs its own primer. Once the RNA primer is made, DNA polymerase extends the primer by adding nucleotides to the 3' end of the growing strand. During lagging strand synthesis, primase synthesizes short RNA primers at intervals along the template strand. DNA polymerase D extends each primer, forming Okazaki fragments. When the polymerase encounters the downstream fragment, the RNA primer is displaced into a 5' flap structure. This flap is cleaved by Fen1, flap endonuclease 1, and the remaining RNA is degraded by RNase H. DNA polymerase D or E then fills the gap with DNA, and DNA ligase I seals the nick, completing the strand. Now, along with these enzymes, there are also some other important proteins involved in DNA replication. The first protein is the sliding clamp. Sliding clamp is a protein that holds DNA polymerase to the template strand. Without the sliding clamp, DNA polymerase falls off the template strand very easily. The next protein is the clamp loader. Clamp loader is a protein that loads the sliding clamp onto the template strand. Next protein is single-strand binding protein. As mentioned earlier, these proteins hold the two strands apart at the replication fork. The next protein is the replicative helicase. This is the enzyme that actually unwinds the double helix. And lastly, there is the MCM complex. The MCM complex is a protein complex that is responsible for the initiation of DNA replication. Now, along with these proteins, there are also some other non-protein factors required for DNA replication. The first factor is ATP. ATP is required by almost all the enzymes involved in DNA replication. For example, helicase uses ATP to unwind the double helix. The next factor is magnesium ions. Magnesium ions are required by DNA polymerase for the addition of nucleotides to the growing strand. The next factor is a primer. As mentioned earlier, a primer is required by DNA polymerase for the initiation of synthesis. The next factor is a supply of nucleotides. DNA polymerase requires a supply of nucleotides to add to the growing strand. Lastly, there are also some other cofactors required for DNA replication. One such cofactor is zinc. Zinc is required by topoisomerase. Another cofactor is manganese. Manganese is required by primase A. And lastly, there are some small molecules that are required for DNA replication. One such molecule is deoxyuridine triphosphate. Deoxyuridine triphosphate is a building block of DNA. Now, DNA replication is very important because it ensures that each daughter cell receives an identical copy of the parent cell's DNA. Errors during DNA replication can lead to mutations, which can cause cancer or other diseases. Several drugs and chemicals used in chemotherapy target enzymes involved in DNA replication. For example, 5-fluorouracil is a drug that is used to treat cancer. It interferes with the synthesis of thymidine, a building block of DNA, leading to the inhibition of DNA replication. Another example is methotrexate. Methotrexate is a drug that is used to treat cancer and autoimmune diseases. It inhibits the enzyme dihydrofolate reductase, which is involved in the synthesis of nucleotides. So this was all about DNA replication in eukaryotes. I hope you found this video useful. If you have any doubts, please leave them in the comments section. Thank you.